Okay, so our next speaker is Professor Antonia Mey, and she's a Chancellor's Fellow and a Senior Lecturer or Associate Professor in the School of Chemistry at the University of Edinburgh. And she will, okay, let's wait a little bit for the slide to pull up, but she has done a lot of amazing work on drug design, and today, yeah, we will hear a lot from her about her work.
Good afternoon, everyone. First of all, thank you very much to the organizers for inviting me and giving me the opportunity to speak here today. Second of all, maybe a bit of a controversial topic. It wasn't quite what was in the program. I think it never made it into the program. So basically, hopefully in the next 15 minutes, 20 minutes, I'm going to talk about what my group's been doing in the space of both sort of generative modeling and machine learning, and how we've been using language models, or how useful language models are in this area. All right, thankfully, we've already had a nice introduction in terms of drug design, so we can do high throughput screening, we can do fragment-based drug design. In the end, what we want is some kind of compound that works well in our target protein. And of course, we can optimize various different properties, ADME properties, but also binding affinity. So we kind of want some in silico screening way of being able to elaborate either a fragment or find a good hit molecule from our high throughput screening. So what are the methods you can do this with? So one of the things we've been looking at is active learning. Active learning particularly looking at sort of compound libraries that are similar, so you can use things like relative binding free energy predictions, and you can iteratively label your library of ligands you want to score and then figure out which one might be the best compound. Another way of doing it is if you have a generative model, you need to somehow figure out how good that molecule is binding into your binding pocket. So typically what we've been doing there is just looking at docking scores, and as we've just learned from the previous talk, they might be slightly dodgy, and we'll see similar results in a few slides. So because we're kind of looking at different ways of modeling things, looking at libraries, but also at generative design, I thought I'll give a brief highlight in terms of what we've been doing in terms of fragment-based design for generative modeling. So this is work done by Nicholas Runchu, who was in my group as a master's student, and things that are being picked up now by Oro Patnaik, who's been working on this. And the idea is that we're using a diffusion model. So typically in a diffusion model, you train on a data set by adding noise, and then you learn the reverse process to generate samples. So we train a model, and then we generate a bunch of samples. Unfortunately, cats can't quite cure diseases yet, so we probably want to sample molecules instead of cats. But still, we have a problem, because now we've just generated a bunch of molecules. Most of them are probably going to be garbage, cannot be synthesized, as we've learned. So this might not be a super genius strategy to actually do things in this way. So is there a way in which we can actually refine our model such that we can generate something we're actually interested in and want? So again, image machine learning to the rescue. We can use things like refinement, so we can actually post-training refine our model to kind of guide it in the way we want it to go. So here's an example of generating caffeine using a diffusion model. So that is fueling the scientist, but still not quite a drug. But it means we have fairly stable control over how we are interacting with the diffusion model and what kind of molecules we can get. So in terms of fragment-based design, what can we do in this setting? So here's an example of two fragments bound to IMDBH. And the question is, how can we link these fragments? So we're using this refinement approach for our pre-trained diffusion model, and we can actually generate linkers quite easily. You could also use a genetic algorithm to do that. But I guess the whole fun part about machine learning is trying new tools and see if they do better than existing tools. So what we can do, well, in this case, we actually took a very conservative sample of 1,000 molecules. That took about 22 minutes, so we're still not super fast with sampling. Then we can do a bunch of filtering, and we end up with 112 molecules that are good in the sense of they fit the binding site, there's no weird strain, they are sensible molecules based on a set of filtering criteria. Now, I don't have a massive wet lab where I can go and test things, but the nice thing is we took this example from the literature, and in these 112 molecules, we actually recovered 2 compounds that were tested in the lab, and they had nice affinity. So one of the things we're trying to do now is actually use this prospectively, not just retrospectively, but at least it's quite promising that in this particular case, we managed to recover something that medicinal chemists had come up with and tested in the lab and had good affinity. So that's kind of where we're at now. So that leaves us with, well, if we can't afford to go in the lab, can we actually use a model that allows us to predict how well our generated compounds or compounds from a compound library can bind? And this is kind of where my background comes in. So I came into all of this from a molecular dynamics angle. So we've been looking a lot at these chemical free energy types of calculations. We're running MD simulations and then try and figure out from these MD simulations how well a protein and ligand binds. So that is a fairly costly method. It works well with congeneric series. If we have non-congeneric series, we learned earlier today there are new methods that are great, but in general, MD takes a bit of time and you might not be able to score, I don't know, 1 billion compounds. So you could do docking. As we've just learned, docking isn't necessarily super reliable. Or you can use machine learning. And now there's the question, are these machine learning models that have been coming out actually any useful? And this is kind of similar in the veins of what Pat Walters has been saying this morning. We can test whether the way we think about it is the same way you think about it in terms of how we should be assessing these models. Because what is happening is we have a large set of data, we evaluate the model on the data, and then we have a nice table and then go, okay, we have a great performing model. But that doesn't really get us to the point where we're like, hey, I have a particular target and I want to know how that works. So here's a non-naming and shaming table you would typically find. And if we're just looking at some of the data entries, so all of these rows are different machine learning models for binding affinity predictions, and you've got high correlation, low root mean square error. But how different are they really? Does the bold number actually mean anything? There's no error bars on this, so we have no way of actually telling whether one model is better than another. They're all evaluated on PDB-bind core set. Well, that's great, but the test data has many different targets in there. So is that really the kind of question we're trying to answer? So if we're trying to answer, I want to pick my favorite machine learning model, I want to score my compound library, and I want to score it against a particular target, am I going to get there with this? Because based on that, I can probably pick any of them, and they should all do really well, right? So how well do they actually do? Well, here's an example of one we've looked at. This is just out of curiosity. If we try and generate a model ourselves, how good does such a model work? How well does it work? So these are 3 variants of the model. We get a high correlation. That's all great. Now you observe here, I say random here at the bottom. So that's a random test training split. That's fantastic, but it doesn't help us with my particular target example. So if I've already seen the target in the training data, then probably I'm going to do quite well. So if we do different splits, like cold target, cold drug, or cold scaffold, you end up losing your ability to actually be predictive. So you need to be careful in which way you actually work with the data. And that is true for all the metrics. And also observe now, because I've put some error bars on these, I can actually distinguish my different models and go like, there's a significant difference between them and whether they are useful or not. Is that the way to go? Probably still not quite. So right now, we've amalgamated all of our protein and ligand binding affinity data. And effectively, what we've done is plotted this plot and worked out the Pearson correlation coefficient. So we have experimental PKD versus predicted PKD. And then based on that, we can work out our Spearman and correlation coefficient. Great, so that's what we've done. Does that help us with the target? No. If we look at one target, we can look at the scatter. That's kind of similar, so that's great. But if we look at a different one, we might actually have a negative correlation, and it's not very predictive in terms of rank. And we might have another one. So really, looking at just the scatter of this is not going to give us anything useful. Instead, we should look at a different way of combining our individual target predictions using a Fisher transform. And that Fisher transform basically allows us to get a much more reliable error estimate on how good our spread is in the data versus just looking at the overall scatter. This is nothing new. In fact, Pat has written a blog post about this in 2019, particularly looking at the sort of free energy community at the time where people were comparing affinity predictions across many targets and benchmarks. And I think this is still true, and we're not doing it for here. So what is the model I've been showing you? Well, this is now the language models. So they're not large language models. What we're looking at is protein language models. So we have protein sequence and then a embedding or a protein encoding that results from this embedding using a language model. And we have smile strings and the same thing. And then what the model is doing is we're basically projecting onto a cosine space and looking at distances between protein and ligand space. That means if a ligand has a good binding affinity, it kind of moves closer to the protein space if you think of it in that way, whereas if it has a bad binding affinity, it moves away. How does this work in practice? So the architecture is using an ESM2 model and a Camberta model. And then we've actually put some parameter-efficient fine-tuning adapters on each of them, so we actually can individually fine-tune our language models in order to be getting better at actually capturing this. So now, what are the different bars I've shown earlier? The parameter-efficient fine-tuned model plus this BAM binding affinity model is the dark bar. This is the baseline model. The gray is basically just concatenating our protein and ligand encodings and using multi-layer perceptron in order to get your readout for the affinity. And then the middle one is just the BAM without the fine-tuning. OK, so we've generated a new model. It's another black box, a black number in our table. Well, I want to know how good is this model in a sort of real scenario. So we now look at a different set of splits. So the training was done on this leaky PDB bind. And now we're actually looking at different target families. So how good does it perform across different target families? And how good does it perform in comparison to docking? So if we learn that docking is random, well, maybe this is also random. We'll see. But effectively, what we now see is the blue dots are the language model, the trained language model. The orange ones is the docking. For this particular family of proteins, then chaperone targets, again, the BAM model seems to be doing quite well, but then there's also, again, examples where actually docking seems to be doing better. So you really can't just put everything into one pot and get a guaranteed working model. They will work differently across different targets. So now, if I want to look at my overall correlation coefficients and root mean square error, I need to average over them, not by just adding them, but using my Fisher information. So now I can look at my Spearman Rho or my Pearson R versus the different tools we're using. So we've done docking with VINA and RDoc, and then the model we've got. Is this model better than these models? I don't know, because we're not looking at the same things. Am I going to rerun all of this? No, probably not. But I think, at least in this way, I've now can convincingly say, I know for particular target classes, this model will probably do quite well. For other ones, it probably will not do well. Is it learning the right thing? Maybe. Something to look into. So the last thing I want to mention is zero-shot learning and few-shot learning. So very briefly, I talked about active learning. So you can use active learning to score here. We were thinking, what if we could use something like this in an active learning situation? So here is a USP7 target that was not in the training set at all. And we actually get pretty reasonable statistics if we look at the scatter. But if we now take 10 or 20% of the data and do few-shot learning, we actually get a much better correlation. The interesting thing is if we now take the same data and train a Gaussian process, we get almost better correlation. So what does that tell us? Well, in some cases, the Gaussian process is better. In some cases, it isn't. Because the Gaussian process requires you to make a decision about the embeddings. And depending on the embedding you choose, you get similar performance to the model we've been training. If you choose a bad embedding, it actually is worse. And you can't know this a priori. So it depends. It's not the same across all targets. It's going to be different across targets. So yeah, few-shot tuning can improve performance. But Gaussian process models perform similar in certain situations. So this kind of gets me to the end. If you're interested in how this works, all of that model is online and can be tried out. It would be great to kind of hear what you think about it. I think in terms of what I've talked about, so we can do fragment linking with diffusion models. Getting meaningful data splits is important. And language models can be used to predict affinities, so kind of like a scoring function. The last thing I want to mention before acknowledgements is if you work in this field and you have interesting best practices, tutorials, lessons learned, please consider the Living Journal of Computational Molecular Science. I'm an editor for it. If you want to know more, please come and talk to me about it. If you do open source software, consider JOS. Another nice journal. Both of these are Diamond Open Access, so non-profit and ultra-low cost for both reader and author. With that, I would like to acknowledge Rohan and Oro, who did most of the work, and my group, and you for your attention. Thank you.
Great. Thanks for the awesome talk. Any questions? You can come to the mics. I have one. We have maybe 2 or 3 minutes. But I have a question on: have you tried with more like decoys? Because I feel like a lot of times we have different scenarios, right? We in the lead optimization stage, we care more about correlation because we want to boost up the affinity. But in the kind of the virtual screening case, when we have a lot of negatives, have you looked at, for example, like the top hit rate, like enrichment type of thing?
So I mean, the data set has both binders and non-binders in it, right? So in that sense, that information is there. We've not specifically looked at this, no. I know there's work out in that direction, but no, we have not.
Okay. There's. Please go ahead.
Thanks. It's a wonderful talk. You at one point differentiated between large language models and, I guess, just language models. Could you elaborate on that a little more? It seems like that there's a lot of resources being poured into large language models, as we know.
Yeah, okay, so I think maybe my terminology was a bit wishy-washy. So I guess large language models, typically we think of ChatGPT, those kind of type of models, Llama, whatever, Gemini, you name it. So that's trained on language and text that is widely available. Here we look at language models that use similar architecture to these larger language models, but are trained on sequence and smile strings. So the idea there is that we have much more information in sequence databases and smiles databases than 3D structural information. So we wanted to see what if we fine-tune based on then affinity data, can we get a signal out, or is it all garbage effectively? But yeah.
And is there a difference then between, say, tuning a large language model on the exact data set you say and starting?
Yeah, so basically so we do 2 types of fine-tuning. So we have these PEFT adapters, which are basically taking the pre-trained model and just optimizing it a little bit in the training process. And then we have this few-shot training where basically we leave most of it intact and just look at the projection layers and unfreeze some weights there and retrain that bit. So there's different parts where you can basically unfreeze and retrain depending on your desires. But yeah.
Thank you.
Nice talk. With these language models and few-shot techniques, I think it's difficult to not fool yourself and just create a really elaborate similarity search. So did you do controls on this? And did you say, great, instead of zero shot, I'm just going to do KNN with k equals 1 and do the lookups and just do the comparisons?
So we looked at molecular weight comparisons effectively. I don't think we have the plots that really show this, but I think we should do that.
Yeah, I mean, I think just doing a simple similarity search comparison and convincing yourself that you're not just creating a very fancy lookup table.
Yeah, no, I think I think this is why we looked at the Gaussian processes, because to me that is already like a slightly fancier version of a lookup table.
Yeah.
And because they seem to work surprisingly well. Yeah, no, that is a concern, 100%.
Okay, thanks.
Thank you. Yeah, with that, let's give our speaker one more round of applause.